AMALearning, you're about to. Specification 0.3.9b. Describe how genetic variants in the non-coding DNA of a gene can affect phenotype by influencing the binding of RNA polymerase and altering the quantity of protein produced. This is a biology-only specification point. To successfully answer any GCSE questions linked to this topic, you will need to be able to describe how a genetic variant or a mutation within the non-coding part of the gene will impact on phenotype or protein production. What is non-coding DNA? Think of DNA as a big instruction manual. Not all parts of it give direct instructions to make proteins. Some sections work more like a control panel, deciding when and how much protein is made. These are the non-coding regions of DNA. What happens when non-coding DNA changes? Sometimes the control panel gets a glitch, called a genetic variant or mutation. If the variant or mutation makes it harder for RNA polymerase, the copy machine, to bind, less protein might be made. If the variant or mutation causes overproduction, too much protein can disrupt how the cell normally works. How common are genetic variants or mutations? Most variants or mutations don't have any noticeable effect and are harmless. Rarely, some variants or mutations can be helpful, and some may be harmful and cause diseases or disorders. Genetic variants can show up in the coding or non-coding parts of DNA. While many don't cause problems, some can change how proteins are made, affecting traits or leading to inherited conditions. Let's apply what we have learnt to a past paper question. In this question, we are being asked to describe how a mutation in the non-coding region of the DNA can prevent a gene being transcribed. This means we must link our learning from this lesson to our learning from specification 0.3.8b linked to transcription. We know from our learning that if the variant or mutation in the non-coding of the region of the DNA makes it harder for RNA polymerase to bind, less protein might be made. We also know from 3.8b that the RNA polymerase uses the coding strand of DNA to make a complementary strand of mRNA, or messenger RNA. If we link these two key parts of our learning, we can understand that if the variant is making it harder for the RNA polymerase to bind, then less mRNA will be produced, and consequently, less protein will be made. If we compare our learning to the mark scheme, we would have got the marks outlined in both bullet points, so full marks. Well done. Our animations were designed by experienced UK-trained teachers. We have been achieving amongst the best results in the country for many years, and we decided it was time to share our success. Have you just AMALed and want to AMAL some more? Don't forget to like, subscribe and share to keep yourself up to date with the latest uploads. AMALearning, you just did.